Hello students, welcome to EPG Pathshala. I am Dr. P.M. Reddy from the Energy and Resources Institute, Delhi. Today we are going to talk on module Ti and Ri plasmids, features and their use as a binary and cointegrate vectors under the paper Plant Biotechnology and Crop Improvement. Learning objectives of this module are to understand the key features of Ti and Ri plasmids, to comprehend the molecular mechanism of gene transfer from tDNA to plant cell, to familiarize with cointegrate and binary vector transformation systems. Outline of this talk is Agrobacterium, tumefacients and crown gall disease, explanation on that, followed by what are the characteristics of Ti plasmid and mechanism of gene transfer from bacterium to plant, cointegrate and binary vectors, which are useful for gene transfer, and Agrobacterium transformation in plants, and Ri plasmid and hairy root induction. Agrobacterium is nature's genetic engineer. This is a gram-negative bacterium which is able to transfer bacterial DNA, which is called the tDNA, into the host plant. Normally, the host plants in nature are dicots. And this formed an important tool in the hands of scientists to develop a methodology for a genetic engineering of plants. This Agrobacterium tumefaciens, after transferring tDNA, the bacterial portion of the plasmid DNA, this tDNA produces different types of hormones and creates irregular growth cell division leading to cancerous growth, which are known as galls or crown gall in dicotyledonous plants. Here are the examples of the crown gall formation in various dicots. You can see some abnormal structures on the trunk of the tree or the stem of a small plant. These cancerous-like growth are due to the infection by Agrobacterium tumefaciens. Agrobacterium is nature's genetic entity, is called because it has special type of plasmid which is able to transfer DNA, donate DNA to plant, and that DNA is able to bring about several changes in the plant crown gall formation. The major characteristics of this plasmid, which is known as in case of Agrobacterium tumefaciens, is a Ti plasmid, tumor-inducing plasmid. This is a very large plasmid, about 220 kilobases, single copy plasmid. It has a tDNA, which is called a transfer DNA. It is flanked by right and left borders. These are about 25 base pair repeat sequences. And within this right and left border are several genes which are present, which are responsible for production of enzymes leading to the production of auxins, cytokinins, and opines. Auxins and cytokines, these are growth hormones which bring about cell division and gall formation. That's why you see the gall cancer-like growth. And opine genes produce different types of compounds belong to the family opine. These compounds are utilized by the bacterium for its multiplication and growth inside the crown gall. This is the basic structure. So what we are talking about is this tDNA is very important for gene transfer, which scientists have utilized and exploited this particular plasmid and are able to transfer tDNA along with our gene of interest into various plants. While talking about transfer of tDNA from Agrobacterium to plant cell, this is mediated by tDNA transfer is mediated by Vir genes present in Ti plasmid and the chromosomal genes of Agrobacterium. These virulent genes comprise of Vir A, Vir G, Vir C, Vir D, Vir E, Vir-F, and so on, several other genes. Vir-A is membrane-bound sensor kinase. Vir-G product is transcription factor, and Vir-C is a host range, confers host range specificity. Vir-D acts as an endonuclease. And Vir-E is another important protein product, is to transfer tDNA from bacterial cell to plant cell. This is the one which forms a protein coating around single-stranded DNA, so that it's not subject to exonuclease activity. Vir-B is a transmembrane complex. Other genes, chromosomal genes involve CHVA, CHVB, and PSCA. These are all responsible for developing the complexes for attachment of bacterial cell to plant. How the infection of Agrobacterium occurs in plant cell. There are certain changes, critical steps, which include within bacteria perceive chemical signals coming out from the plants, which are mostly phenolic compounds, which are produced by plants when the plant is injured. And these phenolic compounds, when they are perceived by bacteria, they induce a set of genes which are known as virulence genes, Vir genes, and produce Vir gene product. And tDNA is being excised from this plasmid and transferred with the help of the products of Vir genes into the plant. Within plant, there are several changes occur. The sites for bacterial attachment and the transport of tDNA into nucleus and integration of tDNA. These are things which happen two different processes. The whole mechanism is like this, which is shown on the right side of this slide, a figure depicting the infection of bacterium and transfer of DNA into a plant. Here I would like you to concentrate on a circular structure on the right side of this picture is called a Ti plasmid, and this Ti plasmid has got a region which is flanked by left and right borders that is called tDNA. This tDNA is the one which is being transferred. What is the mechanism of transfer? This plasmid also has virulence genes, Vir genes like B, G, O, D, E, F, and so on. These virulence genes help this tDNA transfer to the plant. Among these virulence genes or gene products, Vir-A and Vir-G are constitutively produced. Vir-A is the one which senses signals coming from plant, phenolic signals, and when the signals are pursued by Vir-A, it gets phosphorylated, and upon phosphorylation, this one phosphorylates Vir-G gene, Vir-G protein, which is a transcriptional activator. This Vir-G is the one which activates all different types of Vir genes to produce Vir gene products. Among this, most importantly is Vir-D, Vir-E, Vir-F, and Vir-B. These are the critical ones. There are many more, but for our discussion, I'm highlighting this. Vir-D is the one, is a nucleus, endonuclease, which makes cuts at flanking regions, right and left borders, a single-stranded cut. And a single strand of DNA is released from that portion of tDNA segment. That's as soon as that is being released, this Vir-D2 is still bound to this tDNA at the 5' end by phosphate bond. This one is transported along with this. Vir-D2 acts as a guide sequence, guide protein. Transport this tDNA to the plant cell through a channel complex which is formed by Vir-B gene products. This Vir-D2 and the tDNA are transferred through this from bacterial cells into plant cell. At the same time, other Vir products like Vir-E, Vir-F, which are generated in the bacterial cell, Vir-E protein products are transferred to plant cell, and Vir-F is the one which helps transfer DNA to pass through this channel, Virbi channel. Transfer DNA, as soon as it enters plant cell, it needs to be protected by exonucleases. It is coated by or bound by Vir-E protein molecules, so that the DNA is protected, it is not degraded. And this also combines with the factors which are the plant-derived factors which interact with the Vir-E protein. And the Vir-E protein as well as the Vir-D protein have got a nuclear localizing sequences which target this, help this tDNA to be targeted to nucleus of the plant through nuclear pores, channels. And once it enters inside the nucleus of the plant, the tDNA gets integrated randomly into plant chromosome, and it is replicated as a part of plant DNA. This is the mechanism of infection of Agrobacterium and the transfer of tDNA from Ti plasmid, which is in the bacterium, to the plant and integration into the DNA. Here this picture shows greater details of tDNA transfer from bacterium to plant cell. And this picture virtually contains what are the details which are explained in the previous slide, but with the greater details where this attachment sites is shown. These attachment protein molecules produced by bacterium are coded by the genes which are from the chromosome of the bacterium, CHV, B, etc. And also depicted in this picture, the plant interacting proteins inside the plant cell with Vir-E2 gene products, which code tDNA. These are the extra details present in this particular. Based on these characteristics of Ti plasmid, since it's a very large plasmid, it is very difficult to manipulate. There are no appropriate restriction sites to insert the gene of interest, and it becomes, and the large plasmids tend to break frequently, so difficult to handle them and also to transform them into the bacterium. So what is done is, in Ti plasmid, the plasmid is disarmed in the sense the genes coding for the gene products for the production of auxin, cytokinins, opines present in the tDNA portion flanked by left and right border. All those genes are removed by genetic engineering. They are replaced by the sequences of PBR322 vector as well as a marker gene, in this case ampicillin gene, what is shown in this picture. So this particular disarmed vector is transferred back into Agrobacterium tumefaciens. There's another vector which is constructed in vitro. This is much smaller vector, easy to handle, where a gene of interest with a promoter and terminator can be inserted into this gene. There are techniques available how to insert this foreign DNA of your gene of interest into this smaller vector. This smaller vector also contains marker gene, ampicillin resistant gene, as well as PBR322 vector sequences. So this smaller vector, after construction, that is after transferring a foreign gene into this is being transformed into agrobacterial cells. In the agrobacterial cells, this gene of interest, as well as it is transferred to this disarmed Ti plasmid, that's a helper plasmid, by a homologous recombination process, because it has similar sequence, vector sequences, and also the sequence of marker genes. So these sequences align and the homologous recombination occurs, and all these vector sequences along with the foreign DNA are transferred to helper plasmid in between the flanking right and left borders. That's the tDNA. So this tDNA now has, disarmed tDNA has the gene of interest. This tDNA can be excised, is excised from this plasmid and is transferred to this tDNA is transferred to bacterium, and this is called a co-integration vector because this is formed due to integration of an intermediate vector with the disarmed Ti vector, and this co-integration of these two vectors forms a co-integrate vector. This co-integrate vector, tDNA portion, is now can be transferred, which has a gene of interest, to the plant through a process, a mechanism which I described just now in the previous slides. Another type of Ti plasmid-based vectors is a binary vector. This binary vector is much more easier than developing a co-integration vector. That's why most scientists prefer binary vectors for gene transformation. Binary vector, that means it has a bipartite, two different types of plasmid. The Ti plasmid is disarmed, one of the portions of this vector. This disarmed Ti plasmid has where all tDNA is removed from the Ti plasmid, which is called a helper plasmid, leaving the Vir genes in them. This large plasmid containing only Vir genes and some other essential genes, but without tDNA, is transferred to Agrobacterium, back to Agrobacterium. There's another smaller vector which is easy to manipulate. This vector into which this tDNA is incorporated, but this tDNA is disarmed tDNA. All the genes which code for auxin production, cytokinin production, opine compounds, they are all deleted, and a multi-cloning site is suitably incorporated along with the plant selection marker. And this is flanked by this tDNA naturally, right and left borders. So this tDNA, this plasmid, once it's constructed in vitro, it is transferred to Agrobacterium tumefaciens. So with the help of Vir products coming from due to the activity of Vir genes in the helper plasmid, this tDNA is excised and transferred to the plant. This shows in the greater detail of two different plasmids when they are transformed into inside agrobacterial cells. You see, this helper plasmid has no tDNA. It has all Vir genes, and some Vir products are also coded by chromosome of the bacteria. And whereas there's a binary vector which is developed in vitro, it contains right and left border and a selection marker for a plant selection and our gene of interest, which can be cloned into multi-cloning site. This forms a tDNA. So when these two plasmids are present inside the Agrobacterium, Vir gene products coded by the helper plasmid, they are the ones which are able to, like a D2 is able to cut, Vir D is able to cut tDNA, single-stranded DNA with the help of other genes is being transported to plant cell, where it gets incorporated. That's how a gene of interest can be transferred from the bacterial cell to the plant cell. So this is a very powerful tool to transfer the genes, several genes through bacteria to transform plants, to develop plants with the various different traits. The advantage of this binary vector is this vector being a smaller vector with a multi-cloning site and the selection marker for a plant selection, easy to manipulate. And the small size is due to absence of the Vir genes from the backbone of the plasmid. Vir products are supplied in trans. This slide gives a pictorial outlook of agrobacterium-mediated transformation in plants. It gives an overview of this, how the gene is transferred from bacterium to plant, how the plants are developed. Here, the first step is the agrobacterial cell containing either a co-integration vector or a binary vector is the culture is mixed with callus cells of plants, plant calli, and allowed to grow together, co-cultivation which is called, and these bacteria are allowed to infect plant cell for about 48 to 72 hours. After 72 hours, the plant calli are taken out and washed thoroughly with sterile distilled water, then medium, and the plant cells are transferred to callus medium containing antibiotic like agromycin and kanamycin to select only those cells which received Ti plasmid to multiply and grow. The tDNA of the plasmid, which contains a gene, is a selectable marker which confers resistance to the plants to allow them to grow on the petri dish in a culture. Once these cells are allowed to proliferate, later these calli are subjected to regeneration, and regenerated plants are transferred to pots, then taken to greenhouse for multiplication. And this is very easy to develop transgenic crops through Agrobacterium-mediated transformation. In dicots, it's very easy. For monocots, it is slightly difficult in the sense they are not very amenable. What's the difference is dicots produce phenolic compounds which can activate the very genes of the Agrobacterium to help transfer tDNA to the dicot cells. In case of monocots, inducing phenolics are not produced so well. So we need to give the phenolics from outside like acetoquinone. It's mixed with the bacterial culture for the induction of virgins, genes, and this bacterial culture mixed with the calli, in this case is rice calli, and these calli are after infection for about 48 to 72 hours are allowed to grow on a selection medium, and the proliferated cells are subject to regeneration, and the regenerated plants are transferred to hardening and finally to greenhouse for multiplication. The other type of Agrobacterium, which belong to the same family, Rhizobiaceae, is Agrobacterium rhizogenes. It is different. It has got a different type of plasmid, which is known as Ri plasmid, root-inducing plasmid. This tDNA, when it is transferred from this plasmid to the plant, it produces hairy roots in the plant. So this system, Agrobacterium rhizogenes system, is used to produce transformed roots in a plant. These are characters which are shown in the picture here in this slide. Is the Ri plasmid character. It has a tDNA with the right and left border, and the region between these two borders are the genes coding for auxin production, cytokinin production, and oncogenes, opine gene synthesis, and opine synthesis, and so on. And these are the genes which are in natural condition are transferred to the plant, which is induced hairy roots. This particular plasmid is not manipulated, unlike in Agrobacterium tumefaciens, Ti plasmid, where you remove tDNA. This tDNA is written. Ti plasmid is not manipulated. But there is a binary vector developed outside this bacterium, which is much smaller. As I have already described this binary vector, it has the right and left tDNA, disarmed tDNA, where a gene of interest can be cloned with a plant selection marker. This binary vector is transformed to the Agrobacterium rhizogenes, and once after transformation, this Agrobacterium rhizogenes is cultivated. Now, this rhizogenes now has two different plasmids, not only Ri plasmid, it also has the binary vector, both of them together. The Vir gene products supplied by the Ri plasmid are able to excise tDNA both from Ri plasmid as well as from binary vector tDNA, which has its gene of interest. Both the tDNAs are transferred to plant cell with a mechanism as I mentioned earlier, and both tDNAs get integrated into plant genome. The tDNA from Ri plasmid induces hairy roots because it has genes for cytokinin production, auxin production, oncogenes, as well as open synthesis for the using by the Agrobacterium rhizogenes. And the Ti plasmid from binary vector contains a gene of interest as well as plant selection marker. It can be one marker, antibiotic resistant marker, or it can be an antibiotic resistant marker as well as a fluorescent marker, just to visualize or select the transformant roots. So when both the tDNAs are transferred, the tDNA from Ri plasmid induces hairy root formation. tDNA from binary vector gives the trait of the gene of interest produced by the gene of interest to the plant. There are few examples of non-establishment of hairy roots are described in the following 2-3 slides. This particular slide explains hairy root formation from Solanum tribolatum. It's a very simple procedure. In this, an explant is taken, a leaf, and the leaf petiole is immersed into a culture into Agrobacterium rhizogenes containing both Ri plasmid and a binary vector, and left it for a day or two, and it is allowed to form due to the transfer of tDNA from Ri plasmid. Hairy roots are produced, and at the same time, if those roots contain tDNA from binary vector also, it produces a trait of your gene of interest, both things. So hairy roots develop from the petiole portion of the leaf, which can grow and multiply in a medium in the absence of phytohormones, because tDNA coming from Ri plasmid is able to produce both auxins and cytokinins. So these hairy roots formed can be cultured in the absence of phytohormones in the medium. This slide explains a selection marker. Just show a GFP, green fluorescent protein, or this is visual markers. You don't have to, when the roots are transformed, you can very clearly see if it's a red color that's transformed by the RF red fluorescent protein like DS red. If it's transformed with the green fluorescent protein, they fluoresce green. This is done, this slide shows hairy root formation eucalyptus seedlings. This shows the hairy root transformation in Lotus japonicus, where a seedling is cut at the hypocotyl region, dipped into Agrobacterial rhizogenes culture containing both Ri plasmid and a binary vector, and incubated. And after some time, these plants are allowed to form hairy roots. Due to the presence of either GFP or RFP, they can easily select it. If the green roots, that means they are transformed with your gene of interest, along with your gene of interest. If their roots are not green, then they are wild type transgenic roots, or they can be wild type roots. Shows hairy root transformation in Phaseolus vulgaris and Glycine max, in other words, common bean or a soybean. This is done with a small seedling again. Seedling is excised at the cotyledonine node and infected with the Agrobacterium rhizogenes. And after infection on the petri dish or in the soil, they are allowed to grow and to form hairy roots. And since this slide shows the selection marker here is a GUS gene. A gene upon supply of X-gluc produces a green product, glucuronidase. So you can see the blue roots. That means that these are the roots which are formed due to transformation. Advantages of hairy roots. Hairy root system is genetically and a biocentrically stable system. In this system, only the hairy roots are transformed by tDNA, whereas the shoot portion of the plant is not transformed. It's a wild type. is called a composite plant. And these hairy roots can be grown in medium containing no phytohormones and used for the production of secondary metabolites. That's how, if your gene of interest is able to produce secondary metabolites, they can be produced in the culture medium. So students, let's now summarize what you have learned in this module. Today, we try to understand features of Ti and Ri plasmids and their use as binary and cointegrate vectors for genetic transformation of plants. Our discussion was mainly focused on Agrobacterium, tumefaciens, and crown gall disease, and characteristics of Ti and Ri plasmids, and mechanism of tDNA and gene transfer from Agrobacterium plant cell, development of Ti plasmid-based binary and cointegrate vectors, Agrobacterium-mediated plant and hairy root transformation. Thank you.